Hi everyone, my name is Cristiane. I am a Brazilian dentist scientist that's currently working as a postdoc here at Luiz Bertelsoni's lab. And I'm excited to talk to you today about a topic that I wish I had known before I came here to the US. It's how to write an outline. First question, what is an outline? The general definition for outline is a tool used to organize ideas about a topic into a logical order. For research, we have outlined as a written plan of the organization of a paper, including figures, data, and everything that you want to put in that paper in a very, very detailed way. The importance of doing an outline is that it will save you a lot of time and effort to conduct your research and to prepare your paper. With an outline, you and your advisor, you can hone your hypotheses, you can track your results, you can keep track of your data, find the best way to present your data, to include the additional experiments that will give that spicy to your paper, and to actually see if your paper is, your experiments are answering the hypothesis, if the hypothesis is backed up, proved or disproved by your results, and if that figures, that paper, the way that it looks like fits the journal that you want to. So with the outline, you are able to see if the story of your paper flows. When do you start doing an outline? It's highly, put that in capital letters, it's highly recommended that you start your outline before starting any of your experiments. You don't need any fancy tools. You're going to have a PowerPoint in your brain. You need to sit down and write on a piece of paper your hypothesis, the experiments that you want to do to prove or disprove this hypothesis, how we're going to analyze this data, what's the main contribution that you think your paper is going to give to the scientific literature. After having everything down, written down in this paper, you're going to transfer it to a second paper and you're going to organize topic by topic as introduction, materials and methods, and discussion and conclusion. And this is the point. You're going to open a PowerPoint and you're going to start organizing the figures in a way that you envision how the figures are going to look like in your paper. So figure one, I want to show this. Figure 2, 3, 4, 5. And at that point, you're going to say what you want in each one of these figures. How do you want them to look like? And what are the elements? How are we going to analyze the data? Don't expect to have it ready in your first attempt. Very, very good and experienced people, they usually sketch one figure 5 to 10 times before they think it's good. I'm going to show you how we have worked with an outline of the tooth on a chip paper for you to see how it has evolved and how the outline prevented us to go to some kind of dead ends or very, very time-consuming roads. I'm going to share the screen with you so you can see here. This is the tooth on a chip paper. And when we started this paper 2 years ago, we knew that we wanted to have the first figure as the fabrication process of the chip. We wanted to work with a 3D matrix hydrogel, so we need to show the diffusion properties of this hydrogel, how the cells would look like in the system, their morphology, and their differentiation properties inside the chip. It would be a way to validate the chip. Okay, but when we looked at this outline, we were not satisfied. It was the innovation would be developing the device, but it didn't actually answer any very powerful scientific question. So we would do a lot of experiments, spend a lot of money and work for kind of not giving very much good, exciting results. So we decided to work with a more powerful and clinically relevant hypothesis. So we wanted to study the interactions of cells with co-culture of bacteria in a chip, to see in real time how stem cells at the dentin pulp interface, they would respond to co-cultures and vice versa. So we knew that we wanted to have first description of the fabrication process of the chip, characterization of the stem cells at the dentin pulp interface, the live and dead assay, the viability of these cells to validate the device, and we would like to have the bacteria characterization on the other side and the co-culture. And then the differentiation and cell markers for the stem cells of the dental pulp. We had only preliminary data, and most of this data here is mock data, fake. And then when we sit down and say, okay, this is a very exciting and it has a convincing hypothesis, a sound hypothesis, but how long is it going to take us to finish all this? At least 2 years, at least, because it's very hard to make the co-culture. Say, do you want to go down that road? Louise and I asked to ourselves, or do we want to characterize and publish the device first, and then go to more in depth with the characterization of the bacterial co-culture? So we stopped the experiments, the co-culture experiments, and we started to work on the validation of the device and to test against dental materials, to present the device first as a platform to test and to test dental materials in a more physiologically relevant manner. So we worked on the first figures, we worked on the beautiful characterization of the dentin pulp interface. We included the data here. This is actual data that is published. And we characterized the nanomaterials against ISO controls. We did these experiments here, functional experiments. But at that point, we were very satisfied with the final results of the paper. We said, okay, we can put some spicy here. So what are we going to do? We're going to make a movie. What is this? What is lacking here? A movie to show in real time how the cells behave. And only after that, we said, okay, now it's complete. And then we submitted this paper, we wrote down this paper and submitted to Lab on a Chip. What I'm gonna show you, that it started before 2018, but the outline, you can see from January 2018 to June 2018, until the publication of the paper in 2019, we worked with the outlines the whole time. And the outlines were our guide to think about the research questions, to think about the aesthetics of the paper, to think about how we wanted the paper to look like, the best version of our data, you see? So, as a recommendation, I really, really recommend you to read this paper here. This Professor Whiteside is a chemist from Harvard University, and has published more than 1,500 papers, and he doesn't do anything without an outline. And here he has a step-by-step of how he instructs his students to do an outline, and it's basically how Luis does here in a lab. And now I don't know how to write a paper or anything without an outline. So, foi prazer estar com vocês, e eu espero que vocês fiquem à vontade para perguntar, mandar questões, para conversar, tá bom? E que vocês possam fazer muitas outlines. Beijo.